So let's start with this chapter. I mean to say chapter already started, but let's understand the concept of this particular session. So well, you know that you've already studied about the intragenic gene interaction. You've already studied. Yes or no? In the last session, madam taught you the gene interaction and all, right? Okay, Sasidaran said, sir, I will join you by 7:15 as I have an urgent work. What kind of urgent work? See, this is a lecture, right? And this is something important. I don't think so that lecture is, I think there is no such important work rather than attending lecture, right? Yes? Oh, so I think so that is important. Yes, you can go now and please join within 5 minutes. Yes? Okay, great. So let's start with this. Yes, so we are going to learn what we are going to learn in this 2 hours session. We are going to learn about the codominance, multiple alleles, epistasis, lethal gene, then pleiotropisms, along with their examples, and polygenic inheritance. So these are the four important concepts which we are going to understand in this particular session. Okay. Now, in the previous class, you studied about how exactly Mendel worked on pea plant, right? So this is what the simple procedure that how Mendel worked on this. So you can see he removed all the stamen from the purple flowers, and he dusted that stigma from the white flower, stamen from the white flowers, and then the first parental generation will appear, and he noted down all these characters. So that is what you have already studied, and that's why I'm not going to take you in a more detail about that because you've already studied. Now, what we are going to learn, I have told you already. But let me first of all clear that gene interactions. Okay, how many of you are very clear, very sure about the gene interactions? What exactly gene interactions and what are the types of gene interactions? Yes, anyone? Who's going to tell me about it? Before I'm going to proceed with this codominance, just tell me what is gene interactions. I just wanted to know that how many of you know about it, or what is that, because you were already studied. Madam taught you about in the last session, right? So just tell me what is gene interactions. Okay, Sajana said it's the intragenic and intergenic. Okay, so I know that is intergenic and intragenic, but what kind of interaction means what? How gene is going to interact? Yes, I want answer from everyone. Yes, Gopinath, and are you there? Hello, and can you hear me?
Yes, sir. Gene interaction is like it's either between a particular trait, the two alleles interact with each other such that will be shown in the both of the particular trait will be shown in the after as a progeny, like after in F1 generation or F2 generation, both the characters will be seen.
Okay,
There is no dominant or recessive there. There will be a mixture of both, and that is known as gene interaction.
okay. Any other want to speak something on the gene interaction? Okay, how many of you know about the gene, allele, and chromosome? How you will differentiate between the gene and allele? Yes, anyone know about the difference between the gene and the alleles? Because in a gene interactions, allele is very important. Yes?
A gene consists of two alleles, sir.
Okay, okay. Yes, let me write here. The first one is gene, another one is allele. So how you will differentiate between these two? What is gene and what is allele?
Sir, as a gene consists of two alleles, it can be said like a gene represents a particular character and the allele each trait of that character. For example, if like tallness is a character, the
Mm-hmm.
gene,
Mm-hmm.
each of the allele might consist of one tall or one dwarf, or
Okay.
both tall, both dwarf.
Okay. Nice. And okay, that's excellently given by Aman. And well, so let me tell you about this gene interactions. Though you have wonderfully explained, but let me tell you about gene interactions, how you will differentiate the gene and allele. So first of all, you should know that where gene is present. Gene is what? It is a particular segment of DNA. Where DNA present, which it is present in the chromosome, right? So we can say gene is a particular segment of DNA which control the specific trait. But in case of allele is what? An allele is a variant of particular gene. It's a kind of variant. Like suppose I can tell you with a very simple definitions. Okay, so suppose this is a particular chromosome, right? So what happened in this particular chromosome? This particular segment, that is here allele A and here is allele small a, or we can say the dominant and recessive, right? So what happened in these gene interactions? As somebody said that Sarjana said that it is of two types, intragenic and intergeneric, right? So what is intergeneric and what is intrageneric? So you can see here intragenic interaction means what? The two alleles of the gene present on the same locus of the homologous chromosomes, that is modified phenotypes. So that is called the intragenic. That means of if this is A dominant, their recessive character is present on the same chromosome. This is A. So we can say this is a intragenic. Are you getting my point? Yes. And if it is present on the different segment of chromosome, then it is called the intergenic. So this is intergenic interaction and this is intragenic interactions. Yes, am I right? Just tell me fast. Is this clear? What is intragenic interaction? The two alleles of the gene present on the same locus of the homologous chromosome. And in intergenic interactions, the gene are present on the different chromosome and produce a change phenotypic effect. Means both producing the phenotypic effect. The region is that the presence, that their locations where it is present. So that is what entire post-Mendelian genetics, it is divided into two types: intergeneric interactions and the intragenic interactions. So two types is very much clear for you. Okay, so one is let me write here intra and the intergenic. Now here intragenic, as you know very well the differences. What are the type of these intragenic interactions? So in that you have already studied the incomplete dominance. Yes, incomplete dominance is the part of intragenic interactions. Another type we can say there is a multiple allelisms. Multiple allelisms. And the third type, that is called the codominance. Okay, so total three type of intragenic interactions. Now, what are the type of intergenic interactions? So, intergenic interactions includes not only these four, there is a lethal gene also can be considered in the intragenic interactions. But what are the type of intergenic interactions? So, it includes the epistasis, the first, epistasis, oh, let me write here, epistatics. Second one, that is duplicate gene. The third one, that is called the complementary, and the fourth one is supplementary interactions. So today we will complete all these types of intragenic and intergenic interactions. Okay, is this clear to all of you? Yes. Now see, in this type of intragenic and intergenic interactions, madam already completed this topic, incomplete dominance. Yes, yes or no? Incomplete dominance already completed in the last session. Yes. So today we will focus on codominance and multiple allelisms. So here you can see codominance. Now, what is codominance? Let's try to understand in details about the codominance. So, yes, codominance. As I told you, the interactions between the different, alleles. Codominance is what? When both the alleles of gene express, see the name itself indicates, co means together, dominance, right? So when both the alleles of gene express themselves simultaneously in the heterozygous zygote condition called codominance. Yes, that means of both the alleles which are present together in the heterozygous individual, but neither shows dominant recessive relationship nor shows intermediate condition, but they express their trait independently. That is called the codominant alleles. Means they are going to show their effect separately. So codominance means together they are individually, they are going to represent their traits. That is what called the codominance. And here in this codominance, the ratio, what we can say, that ratio is always 1:2:1. This ratio is always there in the codominance, right? So you can see in the definition also, when both the alleles of a gene express themselves simultaneously in the heterozygote condition, that is called the codominance. They are not going to show a dominant recessive conditions, nor they are going to show the intermediate condition. They are going to represent separately. That is what called codominant alleles. Now in this codominant alleles should not be confused with the incomplete dominance, because it will create the confusion between the incomplete dominance and codominance, but you should not be confused in that. Why? Because in this because in incomplete dominance, that is not going to be completely represent like dominated. It is halfly, at least partially dominated, right? But in case of codominant, they are fully dominated, but they are going to show independently. That is what the difference in between them. Now here, as we use the letter to represent the particular dominant characters, so here also in the later case, the effect of one allele is more pronounced. So the symbol used for the codominant alleles are different as compared to the incomplete dominance. So let's see what are the symbols and what are the examples related to this codominance. So the very first example you can see here, like, see, this is the white, this is the red, right? So it is always represented by capital R and the character is in W. So this is R and W means what? White, right? And here capital R and a small r represent. Clear? So when both white and red cross, you will get a rayon color, right? So Rohan, so what this actually represent over here? With a simple example, we can say we have ABO blood group, AB blood groups. This is the best example for codominance. This one is the best example. How? To understand this example, let us first share some informations about the HBO blood group, because we know that what is blood grouping, but we don't know about the AB blood group, ABO blood group. So first of all, we should understand what is ABO blood group, so we can able to understand that how this is this can represent best way for codominance. So you can see here AB blood group. So yes. So first of all, let me tell you about the ABO blood group. What is this ABO blood group? So ABO blood group is what? Group in human beings, you know, it basically of 4 types, and that is blood group A, blood group B, we have blood group AB, and blood group O. That means we have total how many blood groups? Total in human beings, total 4: O, A, B and AB. This is what the general phenotypic that we know very well, that we have total 4 type of blood groups. Means not in the single individual, but in a particular individual, either they can have a O group or a B group or A group or AB group, right? These are the blood group types, right? Now this blood group ABO were discovered by Landsteiners in the year of 1900. Okay, A, B and O. That means of these 2 blood group discovered by Landsteiners in the year of 1900. And similarly, blood group AB was discovered by De Castello and Saitini. De Castello and Steiny. De Castello and Steiny. These 2 are different person, okay, which is discovered the AB type of blood group in the year of 1902. Okay, so these are the discovery of blood group A and blood group ABO and AB. Clear? Another important things. Now here, the plasma membrane of human RBC, you know that we have RBC, that is what, red blood cells, right? So plasma membrane of human RBC has glycoproteins, and that glycoproteins called antigen, or we can say agglutinogen, isoglutinogens. So these are what? Antigens. So you can see here that RBC contain, right? So this RBC contain antigens. That antigen is what? You can see here that either we can call it as a RBC or antigen. Now, the type of antigen is controlled by the gene I, and that's why it is written as capital I. So you can see here the type of gene controlled by this antigen, that is represented by I. So you can see here it is always represented by capital I, capital I. So everywhere capital I is there, right? So it is a capital I, and that's why it is written as capital I. So you should not forget that the antigen is controlled by gene that is called capital I, and that's why capital I is represented everywhere. Now here the gene I has 3 alleles. So can you see here the I gene has 3 alleles, and what these 3 alleles? One is having I IO, one is having IA, one is having IB, right? Can you able to find? Yes, you can see here IO, IAA, IAB, IA and B together, right? So that means of when you find out the types of these genotypes, you will get only three genotype: IO, IA and IB. Yes or no? Yes, these three types. Just tell me. Yes? Okay. Now what happened? Both alleles IA and IB are the dominant to the allele IO, right? That means of out of these three, two are dominant, always dominant. This is dominant, right? They both are dominant to whom? To the I0. Means these represent the dominant to I0, but they are themselves co-dominant. That means of they are sometimes they're co-dominant and sometimes they are dominant to I0. So alleles I0 does not produce any surface antigen, means I0 have no any surface antigen. So that's why it is a none. Can you see here? It is written as none. That why it is none? Because there is no any surface antigen. That means of plasma membrane contain no any antigen in I0, and that's why it is none. It is written as none. Clear? Yes. And you can see here it is written as dominant. Who is dominant? Dominant A and dominant B. Along with that, AB, when they are themselves as a co-dominant, right? Are you getting my point? Yes. And I0 is having no antigen, it is none, so that's why it is a recessive. So you should very clear about it that I0 having no antigen, that's why they are the recessive, and I0A and I0B are the dominant and AB is a co-dominant. This you should know very well, right? Now, another important things, IA produce antigen A. Similarly, IB produce antigen B. That means of in the every person, any type of three alleles may be present, and that you can see it is written as capital I used to designate the which alleles are actually derived from the isoglutinate gene, or the letter L for the Landiston. That means of I stand for it's a isoglutino and that's why it is represented capital I. So you should know that why there is a capital I, because there is a isoglutino gene is there, and that's why it is written as letter capital I. Well, so this is what the general informations regarding the ABO blood group. Now let's summarize this table, what exactly is saying. So actually it is this showing the blood groups, their genotypes and their antigens. So a person with I0 alleles have a blood group O. Yes, have a blood group O. Why I0 have a blood group O? Why? Because they neither have antigen A nor have antigen B, and because of that they have a blood group O and it is a recessive types. Yes, are you getting my point? Similarly, a person with IA means you can see here IA or either IAIO, if the person having such kind of genotypes, their blood group is A. So always keep in mind, if the person having genotype IA or IAIO, that means of the person having blood group always A. They have A blood group. Clear? Yes. Why? Because they have antigen A present on their plasma membrane. Similarly, a person with IB or IBIO alleles have a blood group B, right? And if the person having IA, IB alleles have a blood group AB, because both IA and IB alleles are co-dominant, and as I said that it is co-dominant, and that's why their blood group is AB. So this is what the possibility of blood groups, right? So therefore we can say that the blood group, this type of ABO blood group is also an example of multiple alleles. That means of this ABO blood group is a best example for codominance as well as the multiple alleles. Why? Because three alleles govern the same character. No, here total three alleles are together, and that's why they are the best example of multiple alleles also. Okay, so you have to keep in mind that ABO blood group is such kind of example which can be under multiple alleles, which can be under a co-dominance, right? So this example is more suitable for both type of the alleles. Now here alleles for blood group and blood group A and blood group B are co-dominant, so that when they come together in an individual, they produce blood group AB. So it is characterized by the presence of both antigen A, antigen B over the surface of RBC. So it is decided by the surface of RBC what kind of antigens are present. So this is what the co-dominance, they are expressing themselves, separately. So you can see here, as I told, as the definition itself saying that both the alleles, in the previous slide we studied, when both the alleles of a gene express themselves simultaneously in the heterozygous condition, this condition is called codominance. So you can see here, this blood group A and blood group B, both are the dominant, right? Both are the dominants. Yes, that's why they are the separately expressing their dominant characters, and that's why it is the codominant, which is ABO blood groups. Now the another blood group we have, this is MN blood group. I think so this is new for you. You might have not knowing about it. So a MN blood group. Now what is this MN blood group? So this is another example of codominance. Now the first example we studied ABO blood group. This is the another example of ABO blood group. I mean to say a codominance. So MN blood group. What is this MN blood group? So similarly, Landsteiners and Levin in the year of 1927 discovered this MN blood group system. Okay, so it was discovered by Landsteiners and there is a Levin. Okay, these two men who did in the year of 1927 who discovered the MN blood group type. Okay, this is also example of codominance. Now let's see how. So what happened in the MN blood group system, antigen M and N are represent on the surface of ABC, but antibodies are absent. That means of here in the MN blood group there is no antibodies. Can you see here? No antibodies. Only antigens are present, and that antigen can be either M, N or MN, right? Is this clear? Just respond.
Sir, where is MN blood group seen, sir?
Yes, yes.
Where is MN blood group seen, sir?
Okay, let me tell you. I will tell you details. Don't worry. So till now, just confirm that everything is clear to everyone. Come on, do it fast. Yes. Okay. Well, so all responded yes. Now come to our topic. Now, as I told that in the Mn blood group system, antigen M and N are represent on the surface of ABC, but there is no antibodies are present. So that means of antibodies are absent. Now, three type of Mn these blood groups are found, and that is M, N, and Mn. So antigen M is present in the case of M group, N in the case of N group, and both M and in the case of Mn blood group. So this the person with Mn blood group are called homozygous. That means of MMM and MN respectively. With the people we can say Mn blood group are heterozygous, shown as you can see here. It is clearly indicated in this table that the person, if they have a blood group Mn, M, N, and Mn, so their genotype is MM, so their genotype is MM, NN, and Mn. So their blood group can be either, it can be blood group M, N, and Mn. Now the blood group, this type of blood group, this Mn type of blood group does not have any significance in the blood transfusions, because finally blood transfusion is important, right? So here this type of blood group having no any significance in the transfusions, but it is helpful in the solving the dispute percentage and even the identity of a blood stain in the forensic sciences. These type of blood groups are generally used. So basically it is also a co-dominance, but it is not generally used. It is generally used for the forensic sciences, for the investigating and all purpose. This type of blood group generally observed. Clear? So yes, this is about the Mn type of blood grouping. Yes, clear? Yes. Now, and you just ask your questions, what you want to ask.
Is this blood group related to human blood group or sir?
Yes, yes, it's related to human blood group, but it is generally not referred for the normal laboratory. It is just used for the forensic department. Okay, this is another blood group. Why it is here to understand? Because it is also a type of a co-dominant, because M and N both are separately expressed, and that's why these both genes are co-dominant, and that's why we are studying in under the co-dominance conditions.
Is there a
Clear?
recessive gene for this, sir?
Is there
Both M
any?
and N are dominant. Both
Yes.
M and N are dominant. So is there any recessive trait for this?
Uh, See, recessive trait is not there, and that's why there is something reason. Because of that, there is a no antibody. If there is a no antibodies, then Mn itself, they are the dominant, right? And they're dominant, and there is a co-dominant. No recessive present over here.
So it
Clear?
is not a type of multiple allelism, but only
It is a
codominance.
co-dominant. Yes, it is only for co-dominant. It is not a multiple. See, multiple allele we are going to study separately now. So I will tell you. But see, the first one, ABO blood group, is generally it is coming under both type of, and that's why it said it is recessive, dominant, and co-dominant together. So that's why I just gave an example. Just this is also come under the multiple allelisms that we will study. Clear?
What about Bombay blood group, sir? Is it
Okay.
related to this?
Yes, Bombay blood group you're talking about, it is, generally Bombay blood group, or it is also called H type of blood groups, because this type of blood groups are very, rare, and even they lack the H antigen on their red cells, and they has an antibody serums, and it does not express A, B, and H antigen in the cell or tissue. So I think so that Bombay blood group generally it is a kind of deficient in the expression of antigen H. In that condition, they are referred. Okay. So hope this is clear about the Bombay blood groups. and it is not very common. Okay, so this is another example of codominance. Now let's try to understand the third example of sickle cell hemoglobin is the third example of codominance. Now you know very well what is this type of sickle cell and all. So if you don't know, then let's try to focus on this sickle cell hemoglobin. Now what is this sickle cell hemoglobin? You know that we have RBC, that is red blood cells. So it is, due to certain conditions, what happened, this going to be a sickle type of, so that's why it is named as a sickle cell hemoglobin. So what is this exactly? Actually, sickle cell anemia is a hereditary disease which is basically, found in the tropical Africa, but it is now spreading in American blocks, right? And if it is, as it is spread in the African bloc, whose ancestor came from the, part of that Africa. So this sickle cell anemia, uh, in India especially, we can say it is found in some part of Kerala, not completely, because most of the people, most of the Keralite people, they generally love to stay in out of country. So because of that certain currency, it actually originated from that side, and some of the Indian continent you will get this. So in Kerala you will get this type of sickle cell anemia disease. Some part of it is found in Kerala. So person suffering from the sickle cell anemia, if any person suffering from the sickle cell anemia have a sickle cell hemoglobin, and that sickle cell hemoglobin is represented by HbS. So you can see this is the normal blood cells here. This is the complete circle up, like as you can see here, this is what RBC flow freely within the blood vessels. Why? Because these are the normal blood cells. But in case of if there is abnormal or sickle cell conditions, what happen? Abnormal sickled or red blood cells, so it blocking the blood flow. You can see the structure is like this way. So it is somewhat like sickle, yes. And because of this, it is going to block the vessels to flow the bloods, and that is the condition which called it as an abnormal hemoglobin form a strand that causes this sickle shape. So you can see the shape of the RBC, it becomes sickle. So cross section of the sickle cell, which is generally seen like this, the normal one, and this is the sickle shape. Now what are the example, what are the how we can say that? So you can see here hemoglobin, as I told you that a person suffering from the sickle cell anemia have a sickle cell hemoglobin in their RBC. So if any person suffering from sickle cell anemia, they have capital HbS. This is sickle cell anemia represented by HbS gene. Now its normal allele is, if the person is suffering from, but the normal allele is what? HbA. HbA is a normal. This is the normal, and this is abnormal. Abnormal. Okay. Now what happened? A person with ABA or HbA genotypes have the normal hemoglobin. So you can see here a person, suppose this is sickle cell carrier heterozygous and this sickle cell carrier heterozygous, again same, but this HbS is abnormal, HbA is a normal. So what happen if there is a cross, a parental type cross, the first genotype will represent as it is, and in the offspring there are conditions, like suppose HbA is there, then that means of they are the normal sickle cell anemia. But HbS is there, that means of sickle cell carrier. That means of that person, if the person having such kind of cell, that means of they are the carriers. Okay. But if person having HbS and HbA, it is again a sickle cell carrier. So both these conditions are carrier. But if the person having HBS, these are the sickle cell anemia. So the person having HBS, these are the sickle cell anemia, and the person may die in few days, we can say, right? So you can see the condition. This is what the normal. So this is 1. This is what the carrier. The number is 2, 1, 1, 2. So it become 2. And this is what 1. So the ratio is 1 raised to 2 raised to 1. So it is again the codominance conditions. Clear? So this is example related to sickle cell hemoglobin. So that means of simply a person with having HBA genotypes have normal hemoglobin, but the person with having HBS have a sickle cell hemoglobin. That you have to keep in mind. And even you can see the heterozygous, they are the carriers. Heterozygous means what here? This is homozygous. This is also a homozygous, but these two are the heterozygous. So heterozygous is what? It's a carrier. Clear? So this is also the another example related to the codominance. So this is a third example. We studied total 3 examples. Now there is one more example related to the coat color in cattle. This is also the example of what? Codominance. So you have to remember that example because that example is very important. So codominance. So let's try to understand about this codominance. So in the cattle gene, similarly R. Can you see here in this cattle gene? Yes. Here is R, which is represent for the red coat color, and gene W for the white coat color. So I can write this is R, so it is a red coat color. This is W, this is for white coat color. So what happened when red coat color, what we can say that they are crossed with the white cattles, the hybrid of the F1 generation are. So can you tell me what are the hybrids? Hybrid is called the rayon color with the genotype. What are the genotype? That is RW genotypes. So this is what in the F1 generation all are rayon color with the RW genotypes. Now when this rayon coat color cat comprises, they are further, what we can say, hybrids. What happened in the F2 generation? Red rayon white cattles are produced. So you can see here in the next, this is the F2 generations. In this F2 generations, red color, white color along with the rayon color is produced. So red color number is 1, white color number is 1, and the rayon color number is 2. So it become again 1 raised to 2 raised to 1. So this is another example related to the codominance. So that means of they are, equally dominated, and it is a codominant. Yes, is this clear? So here, the rayon coat color are heterozygous, which shows here that R gene and W gene are the codominant. So here R and W are codominant, and that's why it is a codominance. This is example of codominance. Clear? So this is total 4 example related to the codominance. They are equally expressed in that particular trait. Clear? Okay. Now the next concept, that is called multiple allelisms, or you can say the multiple alleles. Multiple allelisms or multiple alleles. Now what is this multiple allelisms or multiple alleles? Yes. So simply when we're talking about the multiple alleles, a set of more than 2 gene present on the same loci or locus controlling the same characteristics is called the multiple allelisms. Yes, they are both controlling the same character. Like suppose I can say the one gene which is responsible for the coat of the cat. So you can see here that more than two alternate form, or we can say more alleles controlling the same characteristics of the trait. Like we can say the coat color is a one characteristics. So you can see the coat color of the cat is in a different versions. Can you see here? So that means of one particular gene is responsible for having these coat color variations. Clear? This is what the example of multiple allelisms. So more than two alternate form of a gene in a population occupying the same locus, that means of they're present in the same locus on the chromosome and known as a multiple allelisms. Yes, so simply you can just remember that the multiple allelisms means what. Yes, is this clear? Okay. Now, mode of origin. What are actually the mode of origin of this multiple allelisms? Let me tell you, the mode of origin, multiple allelisms or alleles are produced due to the repeated mutation of the same gene. That means of that if there is a mutations happening, that is going to produce a multiple allelisms in the different directions. So multiple alleles show a meristic type of germinal variations, and these meristic variations involve the number or the arrangement of the parts. That means of how they are going to be arranged in a particular parts. That cause the multiple allelisms. So because of that, the origin actually started from there due to the mutations. Now, what are the characteristics of these multiple allelisms? How it is different from the codominance alleles, or how we can say these are the multiple allelisms. So there are certain characteristics through which we can say that this is the multiple allelisms, right? So there are more than two alleles of the gene. That is what the multiple alleles is. For example, I can say 15 alleles for eye color in Drosophila and 3 alleles for blood group in human beings, 4 alleles for the coat color in the rabbit. This is all the example of multiple alleles. What? 15 alleles for the eye color in the Drosophila. You know, Drosophila, the fruit fly, they have a different version of their eye color. I will show you the picture also how the eye color is different in the Drosophila. So in the next slide, I think so that is there. So we will I will show you that how the 15 different alleles of eye color in the Drosophila means they're having more than 2 alleles, and if they have more than 2 alleles, so we can say it is a multiple allelisms. Similarly, we just now saw about the human blood group. So 3 alleles of human blood group, that is A, B, and O. These 3 type of alleles in the human blood group is also characteristics of multiple allelisms. Even we just now saw about the coat color of the cattle or even the rabbit, or that is also the example of multiple allelisms. Now all of them are mutant of the same wild alleles. That means of all these characters of multiple allelism, they are actually a mutant. So all the multiple alleles of the gene that are occur on the same locus of the same chromosome, then it is called the homologous, right? Yes. Okay, so now let's try to understand few examples. So you have to remember this, there are more than 2 alleles of the same gene, and all of them are mutant of the same wild alleles. Some gene locus of the same chromosome, or it's a homologous individual passes only 2 alleles of the gene. So the first example of multiple allelisms is blood group human blood group, that is ABO blood grouping is another that is example, which I'm not going to show you more details about. We have already studied about that, how exactly this multiple come under the multiple allelisms, right? So human blood group, that is A, we have a blood group, because this ABO blood group shows both type of alleles, but all cases of multiple alleles do not show the codominance. Let me tell you how. See, there are ABO blood group in the human beings, and that are A, AB, B and O, right? So these later refer to a glycoprotein substance called an antigen present on the surface of RBC. So people with having blood group A form antigen A. Those with the blood group B, they form antigen B. And those with the blood group AB, they form antigen A and antigen B together. Right? So both these blood group A, B having O antigen, O, which is not going to produce any kind of antigens. Right? Yes and no? Okay. Now, determination of ABO blood group. Determination of ABO blood group. You know how our blood group is going to be determined? Yes. Determination of ABO blood group is also called the blood grouping, or it is also called as a blood typing. So how exactly it is detected that the person having A blood group or B blood group or AB blood group? Because generally it is detected by the serum. How? So actually one drop of anti-serum A is placed on the one end of the glass slide, and one drop of anti-serum B on the other end. That means of this is what suppose this is a slide. Let me draw. Suppose this is a particular slide. You know slide, what is slide? You might have used practicals, right? In the 11th standard, slide, cover slip, under the microscope. Yes. How many of know about the slides and cover slip? I know everyone know. Yes, Anne said, Anmoldeep, Sarjana, great. So only 3 students active to respond. Bhuvanasri, great. I want to be active, everyone. Yes. Anthony, are you there? Kirtini. Okay, great. So this is what, just consider the square here, the rectangle box which is represented over here. This is what a kind of slide. Just consider this is slide. So you have to just take one drop of slide of anti serum A and anti serum B on the slide. So this is anti serum A, one drop, anti serum B on the another drop on the same slide. Now what is the next? Now you can see one drop of blood containing RBC is mixed with the serum. That means first of all you have to keep one drop of anti-serum A, one drop of anti-serum B on the same slide, and then you have to put a one drop of your blood in this anti-serum. What happened? So if and when you put one drop, you have to just mix it thoroughly. So slide, so the slide is slightly moved for 2 minutes. You have to just keep it for 2 minutes, and the presence and the absence of agglutination, clumping is observed. So that you can observe through your naked eye also. Under the either you can use a microscope. So if there is a clumping, clumping means what exactly? Thick masses of RBC are seen. Right? So agglutination occurs only in antigen A, not in antigen B. So you can see here this is a thick mass of clump. So right, you can see the clump mass of the how it is clumping formation take place, but in the anti-serum there is no any clumping, right? That means of the person having blood group A. It is detected as the person having blood group A. Clear? Similarly, uh, if suppose there is a no clumping or see absence of clumping, then what we can conclude? The mixture is clear with a separate cell. If agglutination occur with the antiserum A, the blood group A. And if the glutination occurs within the antiserum B, then the person having the blood group B. As you can see here in the second slide, this is the first slide, just this is first slide. Similarly, this is another slide, I can say. This is a B slide. So there is a no glutination in the B slide in the A antiserum, right? And because of that, can I say this person having the blood group B, this person having blood group A. But in case of AB, there is a no any clumping. There is a no any separate cells. So this is what this both represent the clumping. So agglutination occurs only both antiserum A and antiserum B. So the person having blood group AB. So here is AB blood group. And in case of blood group O, what happen if antiserum, you can see here antiserum A and antiserum B, there is no agglutination with antiserum A and antiserum B. So can we say that this is a blood group O? See here, this is blood group O. Clear? So this is what the 4 type of blood group, and we can use antiserum to determine the blood group. Is this clear to everyone? Clotting. Yes, clotting means what? See, clotting, or we can simply, you can see the clumping or a clot or clump, it's same, you know, right? Or it is something different? Yes, and?
No, sir. First you asked the question. I thought you were asking a question and gave the answer. Next moment you said the word itself.
Okay, no problem. So hope this is clear. Yes, I want response from everyone. Is this clear about how to determine the blood groups in a pathology? This is what the normal technique to determine the particular person's blood group. If you have antiserum A and antiserum B, so with the help of this antiserum A and antiserum B, you can able to determine your blood group itself, because this kind of practical generally there in the 12th standard, I think so, 12th or in 11th standard, when we were in 11th and 10th, that time that this type of blood determination type of experiment is there in our okay. So I don't know about whether it is there in your syllabus or not, but see, this is what the general process which you need to remember. Yes, so anyone having doubt on this, the determination of ABO blood groups? Yes, anyone have a doubt? Okay. Okay. Now let's try to understand at gene level that if a person having in a particular family, a parent having blood group A and A or B, then what will be the blood group of their offspring? So how we can determine this? This is also something interesting. So let's try to understand that how if the person or any family group member having the blood group A, B and O, so how we can determine this? So yes, so it's a very simple. When a man homozygous for a blood group A, if any particular man marry with a woman with a blood group O, or vice versa, like that, so all their children will have the blood group A, right? So how actually we can determine this? So you can see here in this first, it is there also. Yes, total 10 conditions are there. Okay, so let's try to understand one by one. Yes, the blood group of parents and their offspring. So what I said, a man homozygous for the blood group A. A man is homozygous for the blood group A. You can see, marry with a blood group, a woman with a blood group O. So all their children will have the blood group A. So you can see here their blood group, their offspring blood group is A. Clear? This is the father. This O, just consider O is father and O is mother and A is a father. So if A is a father and O is a mother, this A and O is what? Blood group of the parent. Okay. So when this blood group is cross, definitely their offspring will always having blood group A. Clear? Similarly, if a man homozygous for the blood group B marry with the woman with the blood group O, that means of this is second conditions. If the man having blood group B and marry with the female having the blood group O, then their offspring will having the blood group B. Yes, so this is what the possibility. But if a man having blood group B and woman have a blood group O, there is no possibility of having the blood group A and AB. This will never appear in that particular offsprings. Is this clear? Yes. Now another one. When a man homozygous for blood group A marries a woman homozygous for the blood group B, that means of A man is going to marry with the B type of blood group of woman, that means of all their childrens may have the blood group AB. Means you can see which condition is applied. If a man having blood group A and female having blood group, or whom are going to marry with the blood group B, so all their children having blood group AB. Similarly, when a man heterozygous for the good for we can say for the blood woman heterozygous for the blood group B, their children may all have a blood group A, AB, AB and O. That means of which is going to be applied. So yes, this is the conditions which is going to be applied here. So this is what the particular, we can say, if a man having different blood groups, so what will be the offsprings of the blood group of their offspring, that we can determine with these simple charts. So you have to remember this chart, which is very important, that if the parents having both having the O blood group, their offspring will definitely have O blood group, but there is no possibility of A, B and AB. Clear? Similarly, if a woman having blood group O and man having blood group A, the possibility is always A. Similarly, man and woman both having blood group A, then it's a A. B, it's a B. So this is what the conditions which you need to remember. Similarly, this is also the similar conditions occupy. Yes. So any doubt in this? I think so this table is very simple. It is not going to create any kind of confusions. Yes. Or if you feel that this is a confusing, then please tell me. I'm going to explain that. Any confusion in this particular table? Only one thing which you need to remember, that you have to be learn this table. This table is important, and if you know this table, you can able to solve the answer very quickly. Okay. Yes, Sarjana, any doubt in this? No. Okay. Okay. Now let's try to understand the blood transfusions. What is mean of blood transfusions? You know, the blood transfusion is based, between the person of the same blood group. But some inter-blood group transfusion can also be safely done if necessary. So, so this blood transfusion generally in emergency situations, the person can donate their blood. So, who actually are the donors and who will be the recipient of the blood group? That's let's try to understand. So we know that what is important over here, that RBC of the person of O blood group have no blood group antigen, right? So the blood group of this person can be donate a person of all the blood group, means O can able to donate the all type of persons having blood group, right? So therefore we can say the person of O blood group are called universal donor. So you can see here this O is the universal donor. Yes, means O person can donate a blood to the recipient A, recipient B, recipient AB and recipient O, which is universal recipient, right? So sorry, AB is a universal recipient. So this is what the person which is recipient means, the person who required the blood, and donor who can donate the blood. So the person having a blood group O, so O person blood group is called universal donor. So they can donate a blood to the all four type of recipient, either the recipient A, either recipient AB, or recipient B or O. They can donate to anyone. Now similarly, a person's of blood AB blood group has both antigen A and B in their RBC, but no antibodies in their plasma. So such person cannot donate the blood to the person of other blood group, but can receive the blood from all other parts, and that's why they're called the universal recipient. Means you can see here, AB cannot donate the blood, cannot donate the blood. You know the reason why AB cannot donate the blood? As I told you that the person having AB blood group have both antigen A and antigen present in their RBC, but no antibodies in their plasma. And because of this, no antigen present in the plasma, no antibodies present in their plasma, and that's why they cannot donate the blood to any recipient, but they can accept the blood from all the donors. Means AB want blood, so they can take blood from A also, B also, AB also, and O also. But AB want to donate the blood, no, AB cannot able to donate the blood because they have antigen, but they have no antibodies on their plasma. Yes, this is what the simple concept which you need to remember, and that's why they're called the universal recipient. Means they can receive the blood but cannot donate. Similarly, O is a universal donor because they can donate blood to everyone. Everyone means recipient A, recipient B, recipient AB, recipient O. Is this clear? So this is about the blood transfusions, and this blood transfusion basically, generally used for emergency conditions where the patient required blood. So in that condition, this type of protocol is followed to donate the blood for a particular person. Okay, so this is also important which you need to remember, that who can donate the blood, who cannot donate the blood, who can donate the blood. See, AB donate AB donor can only donate the blood to A person and AB person. Similarly, B can donate blood to the B person and AB person. AB can only donate the blood to AB only. So AB, if I am AB, so I can donate blood to similar with the AB blood group person. I cannot donate the blood to A, B and O, but I can donate the blood to only AB person, right? and you know the universal and the donors and universal recipient. So this is what something very simple. Yes, anyone have a doubt in this blood transfusions? No? Everything is clear to everyone? Yes, Anthony not responding. Why? Rahul is also not responding. Rahul, Anthony, Bhuvanasri, what are you doing? See, you have to on your camera throughout the session. No camera off. Sasidharan, is this clear? Please respond it in the group, the chat box, right? And that is access to everyone. So you can use that chat box. Okay. Okay. This is something interesting. This is called the chemical basis of ABO blood group. Means we are going to learn that what are the chemical present on this ABO blood group. So basically we know that antigen of ABO blood groups are actually, it is a protein sugar compound. So basically in that ABO blood group antigen, that means of they have a protein sugar compound. It is a protein sugar compound. So according to Cabot and Watkin, what is Cabot and Watkin? This is the name of the what we can say scientist or researcher. Okay, so according to them, the antigens are carbohydrate. Antigens are carbohydrate group, means they are basically polysaccharides that are bound to a lipid molecule of the membrane of RBC. Means in that what we can say blood group, they have a carbohydrate, and that carbohydrate is going to be bound with the lipid molecules, right? So antigen A and antigen B differ only in the terminal sugar molecule of the carbohydrate. Means they have a different terminal regions of their carbohydrate molecule is totally different, and that's why they are differentiated as antigen A and antigen B. So antigen A, the terminal sugar of carbohydrate chain is modified monosaccharide, and that's why it is a N-acetyl alpha D-glutamine. So you can see here it is written also, right? So it is a glutamine, right? So A having their terminal sugar of antigen present is galactosamine, right? So because of that galactosamine, in this N-acetyl group is attached to at the number 2 positions of the terminal galactose sugars. Similarly, in antigen B, the terminal carbohydrate sugar is unmodified monosaccharide, which is alpha D galactose, right? And that's why it is written over here, it is a galactose. Here is a galactosamine, here is a galactose. Similarly, in AB, you can see in AB, what are the component present? So there is A for galactosamine and B for galactose together, right? That means there is no antigen as we studied. Red cell type agglutination none, and the transfusions except from A, B, OB. You know that A can accept the blood, right, from all, but cannot donate. So as written over here. And in case of O, yes, in case of O having no any kind of what we can say the chemicals or carbohydrate or sugar compound on their plasma membrane, or we can say the outer membrane, and that's why it is written as a none over here. So antibody is present here, antigen A or alpha and antigen B or B. So they are the transfusion to O. Only O can transfuse to O. Clear? So this is a chemical which you need to remember, that what chemicals are present on these the surface of the antigen. So that is A, if the blood group A, they have a galactosamine. B is galactose. AB is having galactosamine and galactose, means together both having different, and O is none. Clear? Yes, this is what the chemical basis of ABO blood group. Let me write here the chemical basis of ABO blood group. It's a biochemical basis of ABO blood group. Clear? Yes, anyone have a doubt in this? Okay, now another one. This is also the part of same, but let's having little difference. As you can see here, that almost all individual possess, H substance. What is this H substance? They are called the antigenic precursor H. They are the antigenic precursor H. So this is the H substance precursor. Okay, and in RBC membrane, this H substance is form of 3 monosaccharide, means basically they are made up of from 3 monosaccharides, especially N-acetyl glucosamine, then galactose and fucose. This is important, which I can write here. It is made up of from 3 important H substance. They are the monosaccharides. Okay, the first one, that is called the N-acetyl glucosamine. Second one having called the galactose, and the third one that is called the fucose. Okay, these are what? These are the 3 important monosaccharides that H substance is formed. Means this H substance is basically made up of from 3 monosaccharides, and these 3 monosaccharide is N-acetyl glucosamine, then galactose and fucose. So antigen A and antigen B are derived from this H substance by the addition of terminal galactose sugar. So you can see how this is the antigen A, antigen B, right? So these both are derived. How it is derived from? Derived from the H substance because of the presence of terminal galactose sugar. So this actually explain why A and B alleles are dominant, because of this condition that IO to IO and the codominant, because the person with the both alleles that having IA and IB have both the antigens, and the recessive gene action is therefore, and because of that the result of absence of enzymes, right? So heterozygous condition, the functional gene IA and IB mask the effect of non-functional IO gene. So this is what actually related to how this H substance and how this A antigen and B antigen are different because of their terminal lactose sugar. Okay, so this is also a kind of chemistry of formation of antigen and antigen A and antigen B. So how exactly antigen A and antigen is formed because of these conditions? Yes, clear about this? Okay. Now this is the second example of what? Of the multiple alleles. So in Drosophila, Drosophila, yes, it is a fruit fly. Okay. So here is a condition showing Drosophila. Okay, let me see the image of Yes, This is the different type of color of what we can say eye color of the Drosophila. So yes, I'm here, I think. Yes. So let's focus on this eye color of fruit fly in Drosophila. So eye color in Drosophila is a what a kind of organism. Fruit fly, it is a organisms. Simply it is a fruit fly. Actually we can call it as a fruit fly is controlled by 15 multiple alleles. Means eye color is basically it is going to be controlled by the 15 multiple alleles, and the normal wild type eye color is red, represented by the alleles W. That means of normal is red and wild is W. So there is an alleles W for white. That means of you can see the short one. So here you can see the plus alleles, or we can say R is a red, W that is white, and a small w, or we can say W, capital W, CH for cherry, then blood, then eosine, then apricot. This is what the different phenotypic condition of the Drosophila eyes. Okay, now so the alleles of other eye color traits are intermediate between these two. So they shows the various type of dominance and incomplete dominance. Means and these important alleles are we can see the cherry one, ivory, then there is a coral, then buff, then honey, blood, eosine, and pearls. So you can see the different colors as you can see here. So total 9 types there, right? So this is 1, 2, 3, 4, 5, 6, and 3 is there. 6, 7, 8. Only 8 is represented, but there is an apricot also there, right? So total 9 type of important alleles, which is represented by this 15 multiple alleles. So this is what the eye color in the fruit fly Drosophila melanogaster. Okay, so this is one example of multiple alleles. So this is, you can see the wild type is red color, then this is a garnet, this is the vermilion, then there is a white eosine, then white apricot, white one, then there is a brown, then purple and sepia. So these are the just 9 one, but there are total 15. So that having that all are controlled by the 15 total 15 alleles. Okay, now the another one is a coat color in rabbit. So you can see the coat color of rabbit. So actually rabbit coat color is represented by 4 alleles, and these 4 alleles, you can see what are these 4 alleles. One is C, then CCH, then CH and C. These are the 4 alleles, and their genotype is represented over here. So this is the dominant, this is the dominant, this is also dominant, and this is the what we can say the intermediate one, right? So this is capital C is a wild type, which is generally brown for. Then CCH is that chinchilla, a black tipped white fur is there. Then there is a Himalayan white fur with the black paw, nose, ears and tail. Albino having white furs. So this is what the different coat color of the rabbit. And the coat of the rabbit may have may be differentiated with the different colors, and these color represent, you can see the agouti type coat color. So what is this agouti? So you can see here agouti coat color. Then okay, this is yellow coat color. Okay, this is something different. Yes, so this is what the another type of what we can say example of coat color in the rabbit is a multiple alleles. Okay, so 4 color, I think so this is very clear that agouti, chinchilla, Himalayan and albino types. Yes, this 4 type you have to remember. Yes, anyone have a doubt in this? Okay, just tell me, is this clear to everyone?
So there are 4 types of alleles here,
Yes,
and that 2 are capital C itself.
capital CC, right? So you're talking about the albino type?
Yes, sir. Even the wild type and albino, it is the same as capital C. So what's the difference, sir, in genotype?
Okay, so in genotype, this one you're talking about? Oh, this one, right? Wild type and albino type, right? Okay. See, let me tell you the difference between this white type and albino type. So you can see white type, that means of it having the coat of an ordinary, which is white, right? Rabbit is referred to as a agouti or full color. Entire body color is a wild type. So it has a dark brown or
So
uniform
not
color.
in terms of phenotype, sir. The color is observed, but in genotype representation, both has C, no, sir. So how do we differentiate in genotype?
Okay. See, genotype here is capital C is there, but here is totally they have two different characters, because the eye color is also going to vary, and the coat color is definitely varying. So that we can consider, right? See, in albino type, what happened, the albino coat totally lack in a pigmentation. That means of there is no any kind of pigmentations on their coat color. Can you able to determine that there is a coat pigmentation on the coat? No, it is completely lacking the pigmentations. The eye of an albino also remain pink due to the lack of pigmentation in the iris of eye, right? So that is for the region that albino is represented by the C. So actually here capital C is written, right? So it should not be capital C, it should be small t. So that this is, I think so, a mistake from the type. This is a typo error. It should be small c, not a capital. Yes, and is this clear? This is a typo error. This should be a small c, small c. Hmm? So hope this is clear. Just you can just cut it. You can write here small c and small c. This is a small c, which
Now
is
clear.
I think so. This is clear. Yes, and any doubt? Hope your doubt is resolved.
Yes, sir. It is clear.
Okay, great. Now let's move ahead and try to understand the another concept of intragenic, that is lethal gene. Now what is this lethal gene? So lethal gene means what? Something which is dangerous, right? So a lethal gene can be defined as a gene whose phenotypic effect is significantly drastic to kill the barrier, right? To kill the barrier. Those having that lethal gene, that is difficult for them to survive. So the normal segregation ratio is modified into 2:1 ratio. So how exactly this lethal gene actually? So lethal gene is going to there are basically of three types. One condition is dominant, recessive, and the conditional lethal genes are there. Okay, now what is dominant gene, what is recessive lethal gene, and what is the conditional lethal gene? Let's try to understand. So dominant means, you know very well, dominant lethal genes are the lethal in the homozygous conditions and produce some defective or the abnormal phenotype in the heterozygous conditions. So dominant lethal gene cannot be transmitted to the next generations because the individual carrying this, what we can say, this gene die. So they lost in the same generations. For example, we can say lethal gene was first, it was discovered in the mouse body color. Then it is also inherited in the sickle cell anemia in a man also. Okay, so these are the typical example of dominant lethal gene. Their presence can lead to a drastic change in that particular individuals. Clear? So it is a dominant. Dominant genes are the lethal in the homozygous conditions, and that going to produce a defective, or we can say the abnormal phenotypic conditions. Similarly, there is a recessive lethal gene. Recessive means what? The recessive lethal gene produce lethal effect only in the homozygous conditions. That means of this is going to produce in the heterozygous condition, but this is going to shows in a homozygous condition. Homozygous conditions. Now in homozygous condition, their heterozygote are normal. That means of the if the person having the lethal gene, recessive lethal gene, then what happen? Their homozygous condition is lethal. It is going to affect on that homozygous, but it is not going to affect the heterozygous conditions. So recessive lethal genes are carried in the heterozygous condition. And the most common example is Tay-Sachs disease. Is a kind of disease where you will see this kind of lethal recessive genes. Even the androcephalus in the mice also, albinism in the corn also. These are the different examples of recessive lethal gene. And the another one having the conditional lethal gene. Conditional, the gene which may be normal to the individual in a particular environment. So environment is a condition, may prove to be a lethal when environment is going to be changed. That means of if the environment is suitable, it is okay. If the environment is going to be changed, that gene going to be a drastic change in the particular individual, and because of that there is a condition of environment, so it is called the conditional lethal gene. So for example, we can say in the poultry, okay, so in the poultry a recessive gene cause feathers to break off. The chicken homozygous, the chicken, poultry chicken, so chicken homozygous for this gene are featherless. Why? Because these are the environmental conditions. If these are kept in the relatively warm environment, they survive. But if the temperature fall below optimum, the featherless chickens die. So this is what the conditional gene which is occupying. Right? So this is what the different type of lethal gene. Total 3 type of lethal genes are there. One is dominant, another is a recessive, and the another one is third one is a conditional lethal gene. Yes, is this clear?
So in conditional, the environment kind of like acts like a trigger for the gene
Yes.
to take place.
Yes, environment play a kind of important role for the changing that. See, as I told, conditional means environment is condition. If you providing a suitable environment for that particular organisms, it is normal. But if there is any changes in that environment, that gene may affect significantly. So what happened? That going to be that particular individual is going to be killed by because of that lethal gene. So that is a conditional lethal gene. Clear? Not only killed, but there is some phenotypic appearance will appear, like there in a warm condition there is a feathers, but in a warmer condition feather is going to be fall off. So that is what featherless. So it is also conditional only, environment like hot temperature. Clear? Okay. Any other? Okay. So this is, I think so. Hope everyone clear about the types of lethal genes: dominant, recessive, and conditional. Anyone having question on this? Okay. So as of now, we completed the intragenic interactions. Now the non-allelic, or we can say intergenic interactions. What I said about the intergenic interactions, you know very well that there are various type of intergenic interactions. Some are, so what I said in the in the starting of lecture, that there are intragenic interaction means the gene are present on the different chromosomes and produce a changes phenotypic effect. So example, we can say epistasis gene, duplicate gene, then complementary gene, there is a supplementary gene. All these are the intergenic interactions, right? So let's try to understand these intergenic interactions. So these are the few salient features of intergenic interactions. So what are these? So the very first one, so let me write here for salient features. Okay, let's try to understand the salient features of the intergenic interactions. So very first of all, the what is intergenic interaction? So interaction produce a distinct phenotype different from the normals, and in this intergenic interactions, gene shows a normal dominance and recessiveness and a short independently. As we can say at phenotypic level, homozygous and heterozygous, like homozygous and heterozygous, like capital AA is homozygous, then there is a heterozygous, so it can be homozygous or heterozygous, BB or BB, like this. So homozygous or heterozygous shows their respective phenotype governed by a dominant alleles. So they are designated by either A or B, right? Even in the normal hybrid crosses, F2 genotype fall into 4 phenotypic ratio classes, and that can be either 6 out of 9 out of 16, or 3 out of 16, or 16 out of 3. So their ratio can be like this: 9 raised to 3 raised to 3 raised to 1. So this 9 raised to 3 raised to means what? They can be either 9 out of 16, 3 out of 16, 3 out of 16, 1 out of 16, right? So this ratio can be vary according to this intergenic interactions. So yes, so let's try to understand how it is going to vary. So here you can see epistasis. Now, what is this epistasis? See, epi means above, status means standing. That means of it is a kind of phenomenon of masking or suppressing the expression of gene by another non-allelic gene. Means it is going to suppress the character. Yes, so you can see here, like the gene which suppress the expression of non-allelic gene, that gene is called epistatic gene. And the gene or the locus which is suppressed by the presence of non-allelic gene is termed as a hypostatic gene. So there is a epistatic gene, hypostatic gene, and the hypostasis. Now what is these three terms? Epistatis, let me write here: epistatis gene, then epistatic gene, then the second one, that is called the hypostatic gene, and the third one, that is called the hypostatic hypostasis. Now what is these three different term? So here, what is epistatis? The gene which suppress the expression of intergenic gene is called epistatic gene, right? And the gene which is suppressed by the presence of intergenic gene is termed as a hypostatic gene. And similarly, the effect of gene get expressed due to the presence of non-allelic gene. They are called the hypostasis. So this is hypostasis, this is epistasis, and there is a hypostatic gene. So the best example for this epistasis is what? You can see the examples are red here, and albinism is the example of epistasis. Yes, are you getting my point? Yes. So based on that, epistatis is of two types. Now what are these two type of epistatis? One is called the dominant epistatis and the another is called the recessive epistatis. So dominant means what? You can know in this condition epistatic gene is dominant over its own alleles, means it is going to be dominant over their own gene, and therefore the effective gene in these heterozygous conditions, the dihybrid ratio of the dominant epistatis become 12 raised to 3 raised to 1. That means of, you can see this dominant epistatis there, you know that very well. The normal genotypic ratio in dihybrid cross is this one. But what happened in this dominant epistatis, this is going to be clubbed, so it becomes 12 raised to 3 raised to 1. So this condition is seen in dominant epistatis. Clear? Because it is going to dominate over its own allele, and if it is going to dominate over its own allele, effective even in the heterozygous conditions. So the example related to this dominant epistatis, we can say cucurbita pepo, you know, fruit color in the cucurbita pepo, a kind of fruits. It is a summer squash where you will get this kind of condition. So it is nothing but it is the dominant epistatis ratio is 12 raised to 3 raised to 1. And how this 12 raised to 3 raised to 1? Because it is going to be dominant over its own allele. So it's initially it is 9 raised to 3 raised to 3 raised to 1. So 9 and 3 is dominant over, so it become a 12 raised to 3 raised to 1. This is one example of epistasis. Another one, so this is what the example fruit, white fruit and yellow fruit. So you can see how it is dominated. So total 12 raised to, as I told, 12 raised to 3 raised to 1. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So this is 12. Then this is 1, 2, 3, 3. This is, and this one is 1. So this is 12 raised to 3 raised to 1 ratio is dominant over here. So this is what the dominant epistatis is. Then there is a recessive epistasis. Now recessive, as I told you that in this condition epistasis gene is recessive to its own allele, right? So epistatic gene can have its inhibiting influence only when it is a homozygous conditions. Means here dominant epistasis is going to be happen in a heterozygous condition, but recessive epistasis is going to be in a homozygous condition. Homozygous conditions. And the example for this recessive epistasis, you can see it is a coat color in mice. So what are the ratios? So the ratio can be, you know, this is the common one of dihybrid ratio. So it is going to be dominant on the recessive. This is the recessive one. This is the dominant, right? So this is going to be dominant over this. So it become 9 raised to 3 raised to 4. This type of ratio is going to appear in this coat color of mice, as you can see here. Agouti and albino. So you can see the coat color also. This is normal, but in F2 it become 9 raised to 3 raised to 4, which is albino. So this is in the F2 generation. Yes, so F2 generation ratio is 9 raised to 3 raised to 4, which you have to remember. So ratio is very important in this what we can say intergenic interactions, which you need to remember. Yes, clear? See here, this is okay. So this is another concept as we studied about the epistasis. Now the next is a epistatic gene. This is the duplicate gene. This duplicate gene is also called the pseudo alleles. Now what is this duplicate gene or the pseudo alleles? So actually this duplicate gene, it was observed by GS Sol in the sphere, or we can say the capsula, a kind of a species. Okay, so it is observed in the capsula. Let me write here, cephrid pores, or it is a capsula bursa pastoris. Okay, bursa pastoris. This is the name of the scientific name of that particular plant, and it is observed in Cephrid's persua. So it is a kind of seeded plant, right? So what is observed over here, that duplicate gene or the factors are two or more independent gene present on the different chromosomes which determine the same or nearly same phenotype. If it determines the same phenotypes, so that either of them can produce the same characters. Means if it is same, then it is going to produce their same copy. So if it is same copy, that means of it is a duplicate, right? So the independent gene do not have accumulative effect. So they produce the same phenotype whether present in the homozygous or whether it is present in the heterozygous. It is going to produce the same kind of, it is going to produce in homozygous as well as going to be produced in the heterozygous conditions. In the both state, they are going to show their phenotypic effect, right? So it is generally observed in the endosperm color in maize. So you can see here, this is what the example. So yes, so it is 15 raised to 1. So this is 9 raised to 3 raised to 3 raised to 1. So in case of duplicate, what happened? This all three, it become 9 plus 3 become 12 plus 3 become 15. So 15 raised to 1. So ratio is 15 raised to 1. Clear? Another one, that is called the polymeric or additive gene. 9 raised to 6 raised to 1 ratio. Now what is this polymeric gene? So polymeric, or it is called the additive. Additive means what? Additions, right? So two independent dominant gene, whether present in the homozygous or heterozygous conditions, have the similar phenotypic effect when present individually, but produce a cumulative, or we can say the double effect when found together. Means additional effect is going to be so, and that's why it is called the additive gene, or we can say additive means extra. So it is going to, additive means extra adding, right? So that's why it is showing the additive effect, double effect found together. And in the dihybrid ratio, it become 9 raised to 6 raised to 1 is obtained in the F2 generation as phenotype produced by the single gene dominance of two different genes are similar. So you can see the heterozygous condition, that this is homozygous, this is hetero, and this is homo. So it is going to affect on the heterozygous condition. So it become 9 raised to 6 raised to 1 ratio. Clear? Okay, now this is another one, complementary gene, 9 raised to 7 ratio. Now what is this complementary gene? See, the name itself indicate complementary means what? You know that there are those intergenic gene which independently shows a similar effect but produce a new trait when present together in the dominant form. So complementary gene were first studied by Bateson and Punnett, Punnett square. Yes, Punnett square very well, right? So this complementary gene studied by the Bateson and Punnett in case of flower color of the sweet pea, sweet pea, that is a Lathyrus odoratus, right? So here you can see the flower is also represented over here. So it is observed in this particular plant. Now here you can see the later is also an example of recessive epistasis, where the recessive homozygous alleles of one type suppress the effect of dominant alleles of the other types, right? Yes. So you can see here, this is 9 in 16, 3 in 16, 3 in 16, 9 in 16. This is the first generation. But what happened after, the suppression or the precursors effect? What will happen here? Precursor 1, precursor 2, it's a purple, 9 purple flower is there. Then precursor 1 and precursor 2 is absent. So in that conditions, 3 white flowers. Precursor 1 absent but precursor 2 is present. So it's a 3 white flower. And both precursor are absent, so it's only 1 white flower. So total how many? Total seven white flower is going to be produced in these precursors. How can? With the help of this complementary gene, because it's extra adding, right? So you can see here 9 raised to 3 raised to 3 raised to 1. This is initial in a dihybrid crosses. What happened? This all 3 is going to be added. So it become 9 raised to 3, 3, 6 plus 1, 7. So it become 9 raised to 7 ratio in a complementary gene. Okay. So why is it so far? Because it is believed that dominant gene C produce an enzyme which convert the raw material into chromosine. So you know the chromogene that actually responsible for the pigmentation of particular petals, right? So dominant gene P give rise to an oxidase enzymes that change chromosine into the purple anthocyanin pigment, and this is confirmed by mixing the extract of two types of flower. When the purple flower is formed, this purple flower formation is two-step reactions, and that is going to be you can see the two-step reactions how exactly it. So there is a raw material A, then there is a gene C which is enzymes which is responsible to make a chromogenes, and this chromogene, because of the gene P oxidase present, that is converted into anthocyanin, and because of that the color is going to be purple. So that is what 9 raised to 7. Now this is the last one, that is called the supplementary gene. Now this is what, so let me tell you, this is what the lub. Okay, this is the name of the plant, lub. So let me first of all tell you what is supplementary gene. Supplementary gene are the pair of intergenic gene in which there are two pairs, so one of which produce its effect independently in the dominant state, while the dominant alleles of the second gene needs the presence of other gene for its expression. That means of one is going to produce their effect independently, another is going to be wait for others to present, then only they are going to be represent. That means of simply in the supplementary gene, suppose there are two genes, okay, it is present in the pair. So pair, as I said, means two, right? Two, just consider A and B. So one is going to produce their effect, one is not going to produce their effect, but it is waiting for others to produce their effect. This is what the supplementary gene. Okay, this is the supplementary gene, and it is nicely observed in the plant lub. You know, lub is this is the name of the plant called lub. Okay, it is only known by only lub. So you have to just remember the lub plant for the supplementary gene. Now lub has two gene, and that gene is called K and L. Just consider it has two gene. I can write here K and L. Okay, this is two gene. So in the recessive state, the second or supplementary gene has no effect in the seed coat color. So dominant K independently produce khaki color. That means of initially no effect. So this produce the khaki color, means this a little bit like a khaki color of, the khaki color. Yes, how many of you know about the khaki color? This color is called khaki color. This is a chocolate and this is buff. Are you getting my point? Yes. So what happened if there is a no effect, that means of dominant K independently produce the khaki color. So this is going to produce a khaki color. Now, while its recessive alleles give rise to a buff color, means dominant give a khaki color, dominant, and its recessive give a buff color. Clear? Irrespective of the supplementary gene being dominant or recessive. But in the dominant state of supplementary gene, and that supplementary gene I just consider as a L, L is a supplementary gene. This is a supplementary gene. Just consider. If it is supplementary gene, then what happened? This supplementary gene change the effect of dominant alleles of the pigmentation forming a gene K into a chocolate color. That means of this K, because of the supplementary gene, this K is going to produce a chocolate color. So this chocolate color is a dominant alleles. This change the effect of dominant alleles of pigmented forming a gene into a chocolate color. And because of that, the ratio is 9 raised to, in the dominant, this is what the normal condition, but this is going to be converted, so it becomes 9 raised to 3 raised to 4. So this is what the supplementary gene, and it is observed in lub plant. So you have to please keep in mind the lub, name of the plant is lub, and khaki, chocolate and buff color, you know the region. Clear? Just tell me, is this clear? Okay, now the next one is collaborative gene. Collaboration. You know the meaning of collaboration? Yes. So collaborative means what? The two intergenic gene which do not only are able to produce their own effect independently, but when present in the dominant state, both can also interact to form a new trait. Means the collaborative gene means what? Suppose there is two K, A and B is a dominant, both can able to produce if they are present in the dominant state. But if they are, when they are independent, they present in the dominant state, but they can also interact for the new species. So interacts means in the presence of both, a new trait can also be formed with the help of these collaborations. Means they can express themselves independently, even in the condition they can come together and they collaborate and they form a new traits. That condition can also be observed in a collaborative gene, and that's what is called the collaborative gene. So the two gene influence the same trait, the interact to produce the phenotypic that does not result from the action of either one gene alone. So the gene interact to produce the 4 possible phenotype condition. That is what the collaborative, right? So the example for this, you can see when none of the, when we can say especially example of coms in the chicken. So this is P and R control the com. So you can see how P and R is a walnut, rose, P and single. So these are what the different type of their com in the hen, right? So this is what a kind of collaborative gene. Okay, so yes, how we can see. So this is com type in poultry, an example of collaborative supplementary gene. That is what PR. So when none of these genes are present in the dominant state, so single com is formed. As you can see, the single coms, right? But so this is what the completely single comed is formed. But in case of we can say P alone is dominant, then P com is formed, and that P com you can see here. This is in case of dominant state, dominant singly in a single dominant state, right? Similarly, if R alone is dominant, so here is R alone. If it is dominant, then there is a rose com is obtained. And if both P and R formed together, then it is called the walnut com, and this is what the called collaborative gene to produce the supplementary effect. Is this clear? Yes. So this is the example of collaborative gene. Now pleiotropic, or we can say pleiotropic gene. What is pleiotropic or pleiotropic gene? So you can see here what is represented over here. It is showing something that are conditions in which a single gene influence more than one trait is known as a pleiotropy. So you can see here single gene means what? This single gene uh, can create or can influence the trait of more than one character. So you can see blindness, then liver failure, then heart attack. This is a multiple trait controlled by the single gene. This is what called pleiotropy, right? Means one single gene is responsible for various trait. It is a pleiotropy. Yes. So hope this is clear. What is pleiotropy? A condition in which a single gene influence more than one trait, it is called the pleiotropy, or such gene is called the pleiotropic gene, right? So what are the example of pleiotropic gene? So there are various example, like cotton in the cotton plant. So in cotton, the gene for one trait also influence the height of plant size, ball, number of ovules, and the viability of seeds. So that means of one single gene is responsible for different trait that is going to be controlled. So it is called the pleiotropic gene or pleiotropism. Clear? So this is what the typical example related to cotton, even the garden pea, then Drosophila, even in human being you can say the different type of syndrome, including the sickle cell anemia and phenylketonuria. These are also an example of pleiotropisms. Yes. Now the next one is polygenic inheritance. Now what is polygenic inheritance? So see, again its name indicate polygenic. So you can see a trait controlled by two or more gene. That means of one single character is going to control by more than two gene, then it is called the polygenic inheritance. So can we say it is opposite to a pleiotropic? Because in the pleiotropy one gene control many character, but in polygenic more than two or more gene control the single trait. That is called the polygenic inheritance. Are you getting my point? Yes. So you can see here the different gene going to control the single trait, means skin of the color of the color code, or we can say the skin color. Actually it is a polygenic trait because here it is controlled by too many gene. So it is a polygenic inheritance. Now this polygenic inheritance is divided into two types. One is called the monogenic inheritance, that is come under the qualitative inheritance. So a single dominant gene influence a complete trait. Single dominant gene influence the complete trait. Intermediate forms are not produced, also termed as a monogenic inheritance. That means of this qualitative, because of the single gene is going to be controlled, so it is called the monogenic inherited. Okay, but in case of quantitative inheritance, means polygenic inheritance, here quantitative inheritance means full trait is shown when all the dominant alleles are present, means different eyelids, even the hair color is also a kind of polygenic inheritance. So each dominant allele express a part or unit of trait, so intermediate form are produced, are also termed as a polygenic inheritance. So monogenic inheritance means controlled by one single gene, and polygenic inheritance means controlled by too many genes. Now here two important examples of inheritance, polygenic inheritance, are kernel color in the wheat and the skin color, human skin color. These are the two examples that you have to remember, the kernel color and the human skin color. So this is what, that is entire gene interactions what we studied today. Okay, is this clear to everyone? Please respond it. Okay, then let's try to solve these questions. A person having genotype IA and IB would show the blood group as, actually IA, it should be IA and IB. It shows the blood group as AB. This is because of what? This is a pleiotropy, codominance, segregations, or incomplete dominance. Yes, tell me fast. Okay, so most of you responded B. Yes, absolutely, it is B, that is codominance. Now the next question, in Mirabilis jalapa is a kind of plant. When the homozygous red flower and white flower plant are crossed, all F1 plant have pink colored flower, means initially it is pink in color. Now in the F2 generation produced by selfing of F1 individual, red, pink and white flower red plant appear respectively in the ratio of, or what will be the ratio in the F2 generation if the red flowered variety of Mirabilis jalapa is crossed with the white flower variety. So imagine what will be the ratio. Yes. Okay. So yes, most of you responded B. Okay, so why it is B? So here you can see, when Mirabilis jalapa, or it is normally called it as a 4 o'clock plant, the inheritance of flower color in is an example of incomplete dominance, which you have already studied in the previous session. So what happens when a plant homozygous for red flower are crossed with the white homozygous for white flower, the plant of F1 generation produce pink flower, which is blend of red and white color. So this indicate neither red nor white flower condition is dominant, right? So when two hybrid F1 plants were crossed, F2 generation plant shows red flower, pink flower and white flower, and that condition ratio is 1:2:1. Clear? So yes, you are absolutely right, that is B. Next question, a gene showing the codominance has, what are the character? Codominance. Yes, absolutely right answer. See, we know that co-dominance, co means together, right? So both alleles independently express in the heterozygous condition. That is what the definition of co-dominance we can say, right? So this is absolutely right answer B. Now another one, a person having genotype IA and IB would shows the blood group as a AB. This is because of what it is called. It is a pleiotropy, codominant, segregation, or incomplete dominance. What? Okay, so yes, it's a B, codominance. This is the answer actually. So what we studied the gene interactions, which is intralocus, or we can say allelic gene and non-allelic gene, or we can say intragenic or intergenic interaction. This is intergenic interaction. This is written as actually it should be intergenic, and this is intragenic. Okay, so that is slightly uh, mistake, so you just have to correct it. Okay, so this is what we studied.